Folks, so welcome everybody. I'm Jeff Kahn. I'm the chair of this committee. I'm going to do a little introduction and public disclaimer and such, but a few housekeeping things first and foremost. One, apologies to those who are sitting behind me. It's really rude and wrong for me to sit with my back to you, but it's a sort of challenge in this room for us to do it any other way so that we can see slides and such. So let me just say that in advance. We're webcasting too, so there are cameras and things, and be aware of that. Because of the number of people, there is an overflow room available where there's also live streaming. That's in 207, which should be just across the hall. There is an open Wi-Fi here in the building. It's visitor, no password required. It should just have to accept the terms and then it will work for you. There's food in the cafeteria, which is on the third floor, so one up from us here. And then, apropos to what we just heard, we need to speak into the microphone at all times when we're making comments so that the folks on the phone, who I'll talk about in a second, can hear, and that the webcast can hear as well. So there are buttons that are on/off. I know the members of the committee are used to this, but that's just for everybody else. Maybe before I get into the public disclaimer, we can do a quick around the room and introduce ourselves, and just very quickly, members of the committee who are here in person, and then we have a few who are joining us on the phone because of travel and illness restrictions. Maybe just say our names. I'm Jeffrey Kahn. I'm a professor of bioethics and public policy at Johns Hopkins University. Alta.
I'm Alta Scherer, a professor of law and bioethics at the University of Wisconsin-Madison.
I'm Lori Strongen, and I'm the founder and executive director of Hope for Henry Foundation, a nonprofit based in Washington, D.C., and the author of a book called Saving Henry, which profiled my experience as the first ever to do PGD for HLA typing to save my son.
I'm Marnie Falk. I'm an assistant professor of pediatrics at the Children's Hospital of Philadelphia and the University of Pennsylvania, and a geneticist.
Alan.
I'm Alan DeCherney. I'm an OBGYN, and I'm a branch chief at the NICHD.
I'm Jeff Botkin, a professor of pediatrics and medical ethics at the University of Utah.
We have on the phone, Anna? Maybe.
Yeah, I'm on the phone. Can you hear me okay today?
You want to just introduce yourself quickly?
Sure. Anna Mastriani, University of Washington Law School.
David Chan? Not on? Okay. Who else is on? Jim Childress?
Jim Childress, University of Virginia. I teach in bioethics and related areas.
Okay, and that's it for now. We'll have others who will join, and then a few others who will be here in person.
Kimmelman.
Oh, Jonathan Kimmelman, sorry. Jonathan, you there? Maybe he's muted. We think he's on? He's on. Okay, I'll introduce him. Jonathan Kimmelman, who's actually on. Oh, there you are. Go ahead.
Sorry, Jonathan Kimmelman, Associate Professor of Biomedical Ethics, McGill University.
It's currently in Berlin. Okay, so let me do the public disclaimer and introduction, which is required of us. So bear with me while I read through this. As chair of the Institute of Medicine Committee on Ethical and Social Policy Considerations of Novel Techniques for the Prevention of Maternal Transmission of Mitochondrial DNA Diseases, I would like to welcome everyone to this open session of the committee's second meeting. This is an open, on-the-record information gathering session. As part of the IOM's process, the committee collects information and materials to consider and discuss as it makes its findings, conclusions, and recommendations. Therefore, I ask everyone to be mindful of the fact that the committee has made no conclusions about anything yet. Comments made by individuals, including members of the committee, should not be interpreted as positions of the committee, the Institute of Medicine, or the National Academy of Sciences. In addition, committee members typically ask probing questions in these information gathering sessions that may not be indicative of their personal views and are not indicative of any committee consensus views. Are there any reporters in the room? Do we need to read that out? Yes, okay, thanks. So, for those who are from the media, it's not a news conference, however, and we will not be entertaining questions from the floor. Reporters who are seeking to talk with members of the committee are kindly asked to touch base with the staff director, Ann Claiborne, sitting to my right, and should plan to do so during a break. Let me now describe the study task and process and our timeline. The task being undertaken by this committee for the Institute of Medicine is to develop a report that will inform the FDA in consideration of review of applications in the area of genetic modification of eggs and zygotes for the prevention of mitochondrial disease, specific to mitochondrial DNA. These techniques, which we'll generally refer to as MRT, include maternal spindle transfer, pronuclear transfer, and polar body transfer, but could also encompass other technologies not currently proposed. The committee will take into consideration the complex ethical and social policy issues raised by these techniques to address the conduct of clinical trials investigating these novel techniques, including the foundational question of whether safeguards such as specific measures and public oversight could adequately address the social and ethical concerns raised by these techniques, or whether those concerns preclude clinical trials. The full statement of task for the study is in the handout that should be available, as is the committee's roster. So fuller descriptions of those who introduced themselves a few moments ago. For those of you who are not familiar with the IOM committee process, this is the second of 5 planned committee meetings. The third committee meeting will include another opportunity for the public to comment on the study via a public comment session on May the 19th. Those interested who wish to provide public comments during the May 19th meeting can register on the study webpage, which is at iom.edu/mitoethics. The last 2 meetings are closed and confidential in their entirety. Before the draft report is released to the public, as is the case in all IOM committee reports, it must go through a rigorous review by experts who are anonymous to the committee, and the committee must respond to this review with appropriate revisions that adequately satisfy the Academies Report Review Committee and the chair of the National Research Council before it is considered an IOM report. The report's expected release is in early 2016. Let me now spend a little time giving you an overview of what we hope to accomplish today and how this will work. So this open workshop will be all of today and part of tomorrow. The format includes formal presentations of prepared remarks from the speakers identified on the agenda, followed by opportunities for committee discussion and Q&A with the speakers and those in the audience. The discussion periods will be moderated by the committee members, as this is an opportunity for the committee to gather information to further inform its deliberations. So that's really why we're here. We're doing that in public, of course, but the goal is for us to gather information to inform our deliberations. The workshop will begin with an overview of the ethical, social, and policy considerations of mitochondrial replacement techniques, MRT, and the scientific background of the techniques themselves. I'm going to go through a quick slide presentation in a few moments so that we're all sort of on the same page and we don't need to revisit some of the basics. In session 1, we will explore the ethical and social implications of MRT. Session 2 focuses on a discussion of the extent to which manipulation of mitochondrial content raises social and ethical issues related to genetic germline modification, and whether the issues raised are similar or different from modification of nuclear DNA. Session 3 seeks to explore policy approaches to other scientific questions that may have attributes that are analogous to MRT and what we can learn from them. Session 4 targets key questions in science and medicine, and Session 5 focuses on scientific, ethical, and regulatory considerations in designing a potential clinical investigation of MRT in the United States. Okay, with that, I covered everything I needed to, I believe. I guess we do slides. There we go. I'm going to try to do this from here, big enough that I think I should be able to read it with my weak eyes, but If not, I'll get up and go to the podium. So I'm going to kind of do a forced march here and race us through this so that we try to maybe get back on time. But in an effort to make sure that we all understand what we're talking about using common terms and such, let's go forward. So we recognize that there are a range of ethical and social considerations in the potential uses of MRT, which include kinship, as you see here. So the considerations about the preference for and value of genetically related children in the context of the existence of alternative approaches to creating children. Of course, in the further context of the importance and influence of reproductive autonomy in the United States. There are issues related to the DNA from 3 individuals without calling it 3 parents, because of course that's a fraught way of describing it, but the implications for kinship on the contribution of DNA from 3 individuals, and social considerations in some of these techniques related to the identity of the individual who would be created, parenthood, and ancestry. Second sort of large characteristic raised by potential uses of MRT is this is first in human research, but it's for the purpose of creating children, a little different than the way we think of first in human research in other contexts, and how to think about the ethics of informed consent when we're talking about a child yet to be created or born, and how to think about consent when we're talking about future generations. That children and the women involved are the bearers of the risk of these techniques, and questions of how to describe what is actually being done in the context of MRT. Is it disease prevention? Is it reproductive opportunity? Is it something else? How do we think about the identity of the child who might be created through these techniques? So is it curative, or is it creating a different person? We want to hear people talk about that, and how to understand sort of allowable risk versus the perceived benefit, and to whom. We're not answering any of these questions. You notice they'll have question marks at the end. A third large sort of set of questions relate to risk. Risk to women involved who are the potential mothers, potential donors of eggs or fertilized eggs. The risk to the embryos that are created. Risk to the offspring that may eventually be born. And the risk, if it makes sense to talk about it that way, to future generations. There are, of course, questions of the moral status of the oocytes and embryos involved in the manipulation of them, and different techniques start at different stages of development, and so that will have an impact and implications for how we talk about moral status, as well as the relative safety of each of the techniques. And we'll hear more from individuals with expertise in those areas in the course of our time over today and tomorrow. There are implications around fairness, equity, and access for what will be obviously a limited technology if it is allowed to go forward. How to think about it in the context of fee-for-service in the way we deliver assisted reproductive technologies in this country, whether it might create increased demand for donors of eggs and their payment, and how to think about access in the context of availability of alternative options, such as adoption of children or egg donation. How to think about downstream applications, and when it is appropriate to allow such a technology to go forward. Should there be a threshold of significant or serious disease such to create effectively an inclusion criteria? Is there an obligation for those who see themselves as at risk to use a technology like this? Would they perceive an obligation, and would that have ethical implications? And what would be the potential range of genetic modifications that could be introduced, in the distinction between treatment or cure versus enhancement, and how to understand that in the use of these technologies? And then lastly on this list, the impact of going forward in the context of MRT, and the acceptance or implication for how to think about the germline modification and nuclear DNA debate, which is, of course, being engaged now with other gene editing technologies. So, to echo that, we are starting with the working definition of what is germline modification from the FDA, which, as you see there in quotes, is human inheritable genetic modification. We have a session and speakers who are going to speak to the question of whether this counts, and if so, sort of is it the same or different from, pardon me, the modification of nuclear DNA, and is that distinction of value, and how to think about controls. So I'm going to go very quickly through this, partly because I'm getting quickly out of my depth when I start talking about developmental biology, but let me just go through the techniques that we take to be on the table, as it were. The first is maternal spindle transfer, MST, and these are pictures that come from the Nuffield Council report from 2012. So thanks to them for very clear and helpful illustrations. And the point, I think, here is that you see the process through which we have. Does this not show up? It doesn't.
You can only see it. It's probably not working on this slide.
Maybe okay, so I won't go up there. So just to point out what's going on here, that we have eggs that are being manipulated is really the point, right? So we've got reconstructed oocytes, which contain the woman who intends to be the mother's nuclear DNA and mitochondria from a donated egg. The egg is then fertilized with the intending father's sperm. So that's the important aspect of MST. You'll see we set this up so that we could sort of go through the potential risks of each of these technologies after a very quick description. So one is the concern about mitochondrial DNA carryover, and you see the greater than signs there, with the idea being some of these techniques have a greater risk of that than others, with mitochondrial spindle transfer living kind of in the middle of the range of that potential carryover. The technical nature of the procedure is complex and sensitive to manipulation, and so there's a higher risk of abnormality in the chromosomes than would be the case in PNT, which I'll describe shortly. Ethical considerations is that we're manipulating and destroying oocytes, right? So we're not talking about manipulation of fertilized eggs. However, embryos that are created this way that are deemed not to be suitable for transfer may, in fact, be discarded. So that raises, of course, its own issues. This is just a quick set of where we are in the state of the science in terms of the use of MST in animal models. As you can see, the models in both of these reports are rhesus monkeys. And you see sort of at the far right there, and under the summary part of the table, some of the carryovers or the take-home messages around the results of those animal research reports. Sorry, third animal report, also in rhesus. Second technique is pronuclear transfer, or PNT. The difference here is that we've got fertilized eggs that are being manipulated, and reconstructed embryos are what is then implanted into a woman's body. So rather than manipulating oocytes, we're manipulating fertilized eggs, right? And that's an important distinction between MST and PNT. As you see here, the risks of mitochondrial DNA carryover is greatest in terms of the three approaches in the context of pronuclear transfer. Easier to do technically than MST, but of course it's in the hands of the individuals involved to determine how well or poorly it works. And we've got manipulation and destruction of fertilized eggs and the ethical issues that come with that. State of the science here, you'll notice that we've got one report from mice one report from human zygotes, 2 reports from human zygotes, including the group from the UK, Doug Turnbull, who will join us by WebEx at some point today. Yes, today. Tomorrow, sorry. Okay. Third, we've got oogenesis and formation of polar bodies. The point being here that we've got techniques that involve polar body transfer. We are reconstructing oocytes in the context of this technique, which is then fertilized with the intending father's sperm for implantation. Okay, we've got state of the science here. Models include pig, mouse. Those are the only 2. So we've got some basic science here from animals as well. Mitochondrial DNA carryover is least likely using polar body 1 transfer. Potentially easier to obtain as a matter of techniques, as they're already floating around in their own cell membrane and can be removed with just a micropipette. We're talking about manipulation and destruction of oocytes in the case of this technique, although just as in the case of MST, the embryos created that are deemed not to be suitable are potentially discarded. So we have that concern as well. Second, polar body technology is polar body 2 transfer, so at a different the second polar body rather than the first. And again, we're talking about manipulation of zygotes, somewhat greater risk of mitochondrial DNA carryover in PB2 transfer rather than PB1 transfer. But again, we're manipulating fertilized eggs, although as before, embryos created this way that are not transplanted may be discarded. State of the science here, mouse models. So we're talking about early animal models for all of these cases, and we'll hear more about the implications in humans as I think we go through the rest of today and tomorrow. So that was my very quick sort of race through. I did that as quickly as I could, and I left out a lot of detail, of course. These slides are going to be available. So for those who are interested in reviewing them more carefully, they will be available for you to do that. Thank you, I should have said this earlier, to Anne and Rebecca and Morgan and Mike for all of the hard work that got us to this point and all of the materials and workshop preparation. So with that, maybe I'll just let Vamsi catch his breath and say hello and welcome, and maybe just introduce yourself very, very quickly. We just said who we were and where we work.
So good morning. My name is Vamsi Mutha. I'm a professor of systems biology and medicine at Harvard Medical School and at Massachusetts General Hospital. Our laboratory focuses on mitochondrial biology. And I want to thank everyone that's here today to help us think through some very interesting and challenging issues.
Thanks. So now we're ready to transition to our first session. So maybe we can ask our panelists to resume their spots, or at least to be ready to resume their spots. And I'm going to turn it over to my colleagues, Alta Charo and Lori Strongen, who will moderate this session.